Cigarette smoking kills about half of its users and is linked to various health conditions. These conditions include 17 different cancer types, cardiovascular and respiratory disease, and dementia, such as Alzheimer's disease. In fact, smoking is linked to 14% of the cases of Alzheimer's disease, which is 3 times more than those caused by mutations in any single gene. To continue, approximately 1.3 million non-smokers die annually in the world as a result of diseases caused by their exposure to smoke, which is also known as secondhand smoke. But have you ever wondered what happens to the smoke that lingers after a cigarette is put out? This residual smoke, also known as third-hand smoke, is an emerging field in science and public health and poses hidden dangers that most people are unaware of.
While the harmful effects of smoking and secondhand smoke are well documented, third-hand smoke is often overlooked because it involves low-dose exposure over a long period of time. This is particularly dangerous for children and pets, but also for adults. Nicotine from third-hand smoke settles on surfaces like walls, furniture, and clothing. When exposed to the air, nicotine reacts with other chemicals, forming harmful compounds. One of them is called nicotyrene, which is formed by the oxidation of nicotine. Unlike nicotine, which has been historically linked to addiction, nicotyrene is a carcinogen, as are many other chemicals from third-hand smoke.
To better understand the impact of chemicals from third-hand smoke, we use Swiss Prediction and SuperPred, 2 tools that help predict the biological targets of various compounds and identify the genes and pathways that are likely to be impacted. By analyzing the predicted targets of individual chemicals from THS, we identify several genes that could explain findings from population-based studies that examine diseases caused by third-hand smoke exposure. Our findings validate many of these published results and, for the first time, identify gene perturbations that, to our knowledge, were previously not connected to third-hand smoke exposure. Some of our results point at third-hand smoke chemicals causing perturbations in neurotransmitter signaling in the nervous system, the functioning of the ion channels in the brain, cell cycle alterations, and epigenetic changes that are known to modify the complexion of chromatin, which contains our genetic material.
A major finding from our research is that several components of the epigenetic machinery may be impacted by third-hand smoke. Specifically, we found that histone deacetylase 4, histone acetyltransferase P300, a lysine-specific histone demethylase, and the NAD-dependent deacetylase HER2 and 2 were affected. But what are epigenetic changes? Epigenetic changes refer to reversible modifications of the chromatin, which alter gene expression. Examples of epigenetic changes include DNA methylation, in which a methyl group is covalently attached to cytosines in the DNA, and histone post-translational modifications, such as acetylation and methylation, in which acetyl and methyl groups are covalently attached to histone amino acids. We also found that the progression of the cell cycle is disrupted by chemicals from THS. Collectively, these effects can disrupt the normal functioning of the cell and lead to diseases such as cancer. Moreover, we found that THS changed the expression of genes involved in the functioning of the nervous system and the immune system.
In summary, our research highlights the hidden dangers of third-hand smoke and provides an explanation for adverse effects that were discovered in population studies. Our finding that nicotyrene has multiple biological effects open serious concerns about the underexplored health impacts of vaping and e-cigarettes. By understanding these hidden dangers, we can better protect public health and develop strategies to minimize exposure.